Okay, let's begin. So again, thank you folks for connecting to this second webinar post MPS. I heard you had a blast at this conference. I believe it was an amazing moment where we could see as exhibitor more and more pharmaceutical, dermatocosmetics and nutraceutical companies coming. And the idea of this webinar is to wrap up a little bit on what we show on our booth and both on the different posters and presentation we had. So I'm Thibaut Hanegger. I'm the CEO and co-founder of Netry. I'll be your host today, and we'll have approximately 30 minutes of presentation, and then we can switch to Q&A. So just to wrap up about Netry, we are a French-based company. We are in Lyon. We have 200 square meters of production facilities where we produce our devices and we exploit them with our cell culture labs. We have more than 100 clients now in the world. We've been 5 years old, the company. And we are a team of 34 people. So the idea behind our technologies, again, I'm not going to go into what is MPS, etc. You all know that. But I just want to focus into an application or a specific use cases. So we want to compartmentalize cells to recapitulate connection between cells as all MPS companies. And we do that by having two approaches. The first one is to make compartment and to separate cells using microchannels in which only neurites can sprout, neurons between axons or dendrites, and using microchannels that are 3 micron high. Those compartments where you can see neurons, there are 3 of them, are inside this individual chip that we call the dual-link in this specific case. And all our devices are designed to be compatible in a 96 well plate alignment system. As you can see, the minimum industrial form factor that we have is the quarter bento that is inserted into the neo-bento system that is composed of 4 quarter bentos that are individual and can be, in this case, they can taken out of the system. This neo-bento format is compatible with all lab automation system for pipetting or recording of images. We also provide 3D structures, and I'm going to detail that after, by using nanoporous membrane in this particular case, in this multifluidics line, and that is always into this 96 format alignment well. So let's go in details about those 2 different type of chips. The neurofluidic culture can perform 2D cultures separated by microchannels I mentioned here. So this you can see sensory neurons that are cultivated on the soma side and neurite projecting on the other ones, which gives you access to either cell body or middle axons or end termination axons in a fluidic isolated way, meaning that you can place a compound here and you'll on the right side, and since it's fluidic isolated, the compound will not be seen by the somas by diffusion or by biological diffusion if there's something that's going on from the axons to the cell body. So you can play around placing viruses, RNA, cells even in this compartment to see how they interact with the synapses that are created here in this culture. We have several types of microfluidics organizations. So as I saw before, the dual link with the symmetrical in the middle channel for enhanced fluidic isolation, or to perform an axotomy, for example. You have this dual link shift that is where the middle channel is shifted, and this one is interesting to make pure synaptic connections in the middle channels, because it happens that dendrites will not push more than 150 micron into the microchannels. So we place this little channel 100 micron separated from the channel 1 here, so that you have a lot of dendrites on one side and a maximum of axons on the other side. So in this particular case, for example, for Huntington disease, you're able to make cortical striatum connection in a directional way. We also have the trial-link devices in which you have a much bigger channel in the middle, which allows to place, for example, different cells in the middle channels that can only see the axons themselves for nerve regeneration, or to place Schwann cells. This is the ideal case. We also have options, group system that enhance projection from one side to the other one. All those devices are compatible with the Neobento format I showed you before. So there's no equipment that you require to use those devices. It's a standalone. It's gravity-driven flow. You don't need any pumps. And they are provided in a light version or in a full version. And again, you can take out one of the quarter Mentos anytime you want without displacing the cell culture. And you can replace them by this little plug with DI water in the outside that will prevent evaporation into the entire system. So the system is, we designed the system to be as agile as possible to match your expectation of the cell culture level. Now, all those devices are compatible with standard readouts, LCMS, ELISA, immunofluorescence, as I showed you before. Since all our devices are coated with a 170 micron thick PDMS layer, you are able to make high resolution fluorescence microscopy, both into standard microscope, but also into a high throughput microscope like Opera from Revity, or a Sartorius system, or Incusight, or all of the systems. And we provide the site maps so that you can spot directly when you plug that into your favorite software, so that you don't have to make all the plates map. Now, that being said, a couple of examples. One of example that we made with CIPN, so chemotherapy-induced pain neuropathy, with sensory neurons on one side, the axon on the other side, and we've shown that using paclitaxel or oxaliplatin at several concentrations, we are able to look at the percentage of axons that are damaged, either on placing paclitaxel on the neuron side or oxaliplatin on the soma side. So you can see high-level imaging quantification possible with classical imaging system and software, Imagi, or even software with that are compatible with the most classical computer vision softwares. Now, I introduced before the 3D vascularization system. So those duplex cells, chips, are composed of one opening chamber that is in red here and in here in yellow, which is an open chamber, so you can make liquid interfaces, and that is coupled to a microfluidic channel underneath, in which you have a nanoporous membrane in separation. Nanoporous membrane can be classical 0.4 micrometers nanopores up to several micrometers if you like, and this allows a connection between the reservoirs in blue here into this microchannels that allows to monitor the shear stress on the cells that may be placed underneath. By doing that, you can place endothelial cells in the microfluidic channels and make a microtube all around the channels and expose to the opening here. As an example, we were able to place endothelial cells and human keratinocytes on one side in the well, and obviously endothelial cells on the lower level. And you can see that there's a morphological difference when keratinocytes are exposed or not to endothelial cells, showing that there's an interaction without any fluid leakage. Those devices are fully compatible with TIR system from our partner WPI. Another example that we showed of the usage of the duplex is an enhanced culture of 3D brain organoids. In this particular case, with our partner at CA and Subiotech, we've shown that using those devices, you can enhance the reproducibility of the cell culture by measuring the growth rate of a lot of different midbrain cerebral organoids. How does it do? How does those? Because we will never change the media that is in the brain organoid compartment. We will always change the media from the outside chambers in blue. That means that there will be a natural perfusion of cell culture media into the brain cerebral organoid without touching them. So that will enhance reproducibility because you avoid any shear stress on the cerebral organoid itself. And we've shown that there's much enhanced reproducibilities of cerebral organoids and by extension of other type of organoids. Now, what the next generation of devices that we are currently working on will be the duplex system coupled with the microfluidic system, with the microchannel system. So we both combine innervation with the microchannels, with this cut side, and vascularization, which is with the duplex system. Those devices will be available for purchase in September. Now, once we've done that, innervation of any organs, we are looking at multi-organ system. The way we see and the way the philosophy behind the NETRIS devices is to never use complex pumping system with tubes, with parasitic pumps, etc., because we want it to be as compact and high throughput as possible. So we have developed this patented jumper system that is a double valve system where liquid will be aspirated from a lower compartment, a blood vessel type compartment, aspirated with air and push again to the other compartment with air. So this little guy, it is inserted into the different organization of the duplex and is connected to this, let's say, pumping system that only suck air or pushes it. And this system is fully automated for up to a month into an incubator, making the system totally compatible with high throughput culture and perfusion of multi-organ system. So you can place 1 or 2 or up to 4 organs connected with this blood vessel without any loss of fluid. Again, if you are interested in those applications, feel free to contact us. We are entering beta mode in a couple of months, so if you want to try them, feel free to contact us. So our multi-flake lines is available for purchase into the duplex and the duplex running in a couple of weeks, and always into this neo-bento format in a light off or full version. Now, this was a wrap-up about the devices and what we showcased at MPS. Now I wanted to focus into our initial core value, which is to use neurons as a sensor. And that was the presentation I gave on behalf of Drew at MPS. So the idea behind using neurons, and obviously you have shown you before that neurons go well into our devices, is to couple those compartmentalized devices with multi-electrode array system. Multi-electrode arrays have the capacity to record electrophysiological recording of neurons. And as you can see, with the spread out of axons of multi-electrode arrays onto the entire chip, we're able not only to record onto the cell soma itself, but also into the microchannels themselves. So we partner with our very friends and partner Axon Biosystem to make our devices compatible with the Axon Maestro system. That is the only device that is able to capture up to 752 electrodes at once into our system. So we provide the exact same configuration of our devices into multi-electrode arrays, dual link shift, and trial-link through the entire cell culture system, spread out electrodes. Both versions are available. The Edge version, which is only the upper one, or the full Pro version, with up to 16 chips into the same device. Now, that being said, we will also continue our efforts to use multi-electrode arrays into the 3D system, so the duplex link, by connecting electrodes to the neural channel inside the blue channels I can see, which is connected to the 3D system. And our ultimate goal is to go into a fully automated, portable ADME system towards oncology. Now, today we have explored a lot of innovation capabilities of our devices. So the 2D neurofluidics dual-link MES system is available for purchase. And a couple of examples here with only sensory neurons. When you place a compound into one compartment only, you're able to record for the entire spread out of the cell body, even the microchannels. And you can see, as in the raster plot here, timely recovery of the electrical activity in channel 3, rather more succinct recovery activity in channel 1. I showcase here our gluta and GABA neurons co-culture system with the raw data recording here, and you can see the synchronicity of the GABA system influencing of the gluta system influencing the GABA system through the entire device. And again, you can place, for example, A-beta 42 only onto the GABA to look at how the retroaction loop will interact and change the electrophysical recording if you want to place an anti-Alzheimer drug. We have several use cases also and paper published, for example, in oncology with pediatric glioblastomas. When we place some activated glioma cell lines into one compartment or the other one, and to see the interaction with neurons, and especially when you place a drug that have the targets to reduce the glioma cell lines, and to look at the effect on the cerebral activity. We've shown especially that there's an enhanced activity when the glioma cells are activated or not. Also into immunology, when we place some activated immune cells on, in this particular case, the synapses only, where we show that there's an enhanced activity of cells when the synapses are located. And also we go to a more and more co-culture system. For example, in this particular case, our sensory and primary keratinocyte for this innervated skin model, but in 2D. But also we are opening this new line with an open well, an explant, where you can place an explant system that will be able to connect to microfluidic channel neurons. This particular devices can be used in a lot of different applications for explants, for example, retinal applications, explant of skin, explant of gut, while maintaining innervation. Just to mention also, we are able to place those cells onto a hydrogel or 3D reconstructed hydrogel to mimic the interaction of 2D and 3D cell culture with the innervation that may be able to come from the bottom side. So this chip is available for purchase. Please contact us if you like to have some samples. Now, a little bit more about the methodology behind the recording of the electrical physiological response. There's two ways to record how neurons will interact with the drugs. The first way, what we call slow acting, is a response method that when we record through an entire spectrum of time, that can be up to 24 hours, 42 hours. We've done that up to 5 days, in which we record an altered state that results in a percentage of change in the mean firing rate, which is one of the variables that we can monitor into our system, relatively to reference state. So basically, you have a baseline, you induce a compound, so acute or chronic exposure, you can add on, and you can look at the effect on the neurons activity in a single or a co-culture system. The other methodology we have developed is the fast-acting response method, in which we look at directly when we apply the drug directly into the system while applying, this method allows us to look at both the vehicle, the compound, and have a positive control directly into the same culture, and to even leave it alone to go into slow-acting method after. And in this particular case, we look at the results of an expression of fold change, the mean firing rate, relatively to the reference state. Let me give you some example. For the fast-acting method, we have done a couple of modulators, and recording the changes and the fold of change, when they apply the compound relatively to the vehicle, and to the positive response also. So you can see, for example, paclitaxel I've shown before. There's a significant change when you place a different amount of those response. The capsaicin response also when you place relating to the vehicle or the caps itself, propionate and ATP. So those timeline for experiments, it's up to a month. In a month, you can get all the results, since you are looking at a direct response when you apply the compound. Into the slow-acting method, for example, so we've shown that the temperature has when you apply a fluid on the neurite only with different temperatures, you can really look at the increases of the mean firing rate relatively to the decrease of the temperature. So we crack the code of the temperature encoding into sensory neurons, and propionate, MPS, and lidocaine in specific pain. So this is typically the type of graph that we can provide for a study when we expose only the neurites to a specific compound, and look at either the direct activation or the long-term activation. Now, I show you a couple of examples using one kind of metrics that we can extract, which is the mean firing rate. But with all the variables that we can extract from a complex neural system, PNS or CNS, I detail all those metrics into this slide. We can go up to 42 with specific metrics into the CNS, and also looking at where do you record those metrics into the different channels. So you can see the problem is beginning to get complex in terms of variable to integrate and where you are looking at. So let's go into another method that we've developed, and to look at the altered states that we measured compared to the basic reference states that we disturb while applying the compound or an injury. What we are going to go is that we're going to plot all those different metrics I showed you before into a 2D plot, as you can see, and only into channel 1, the recording of the electrodes that are placed into channel 1. When you do the same thing with all the different channels and the combination of the channels, you are beginning to get a 42 by 12 matrix that will give you a specific image of both cell bodies, nerve axons, and endings of axons. And this gives us what we call a QR code and what we call a digital signature of an altered state versus a reference state. The altered state can be induced either by an injury molecule or a compound to be tested. And those QR codes give us specific insight on how the neuron will encode a response to a remote action of a component. Again, the actions can be either without any cells, so just the sensory neurons themselves, or the innervated organ. Now, when you look at all the different metrics that you can record into this matrix, you can see that some of the variables are in direct combination together and have a very big correlation. So when you apply threshold on this correlation, you are able to extract the variables, mean time reg, synchronicity, or others, that actually describe the state without having any correlation. And when you do that, you extract, in some case, 70% of the variables, which makes it much easier to visualize and to have the sense of what can be discriminating from one signature to the other one. So, for example, in this case, I've shown you that with two different variables that we are looking at, we are able to discriminate only by the digital signature of the neurons two different states of a compound in the blue and the yellow one. And if it's not sufficient to discriminate, you can go into even a hyperplan in which you have more than two new variables. You have much more. You can see the digital signature will have a shape that will give us an insight on what, if we have a specific compound that we don't know the mode of action, we can compare this digital signature relatively to the reference compound that we have in-house. So this is a very new way to have readouts into multiphysical system, and using neurons as a sensor give us a specific insight. Since all the organs are innervated with specific biomarkers at the endings, we anticipate that we can use all those methodologies and extraction of how the neuron will interact to a compound within the organs to give us a global overview of how the organ is without stopping the culture and in a dynamic way. So to put it in a nutshell, I hope that I've shown you the global overview of our unique selling proposition, which will be, first, we develop high throughput and interoperable solutions by using our NeoBento system that is 96 well plate ANSI SBS compatible format. So ready to use without any pumps, without any equipment to buy, apart if you want to go to MEA. And you can automate that into your favorite lab automation cell culture or visualization system. Now, we use the neurons as a sensor to look at the digital signature that is given by a specific compound or molecule reference that is on the market or failed in clinical trials. And since all the organs are innervated by PNS or CNS direct or indirect way, we are able to foresee where all MPS microphysical system will be using neurons as specific markers and biosensor of the state of an organ versus an altered state. And if you push the boundaries a little bit more, and that we do with most of our clients, that will give us and populate their own digital library with a specific model, where you screen lots of different molecules that have been that they know the effects, and we screen those molecules using our high throughput technologies, and we make this digital library for specific indication and a specific model, when you can screen and foresee all the different impacts of a new compound that people want to try, and with a certain percentage of match towards a compound that we know with this specific effect. I hope this was helpful to understand what we do. I've been a little bit short than expected, but I mean it's good for everyone. If you have any question, please feel free to use the chat. I'll be more than happy to answer them. And you'll have a replay of this webinar that you can, with pleasure, push to your teams so that they can get knowledge to what we do. And please feel free to contact us anytime if you have a specific project or co-development project you want to screen. Thank you so much for your attention. Okay, so we have a couple of questions here. So how many cells do you use in your devices compared with conventional methods? So we recommend to use 10,000 to 4,000 cells per channel, so depending on the application, imaging or MEA. The volume of media is approximately 100 microliter per channel. So for one plate, you expect to need between 320K or 640K in only 5 milliliter of media. What we usually do is one full neobento uses one vial of 500K of our sensory neurons. Okay, so would other type of neuropathy be possible to study with your platform? So yes, indeed, we can definitely study a lot of different neuropathies. Somewhat is currently ongoing with a couple of clients and with our internal R&D. We could imagine to study inflammatory neuropathies, diabetic neuropathies, or conditions like CIDP, which is chronic inflammatory demyelination polyneuropathy. And as I've shown you, we use CIPN. Okay, so concerning your CIPN platform, can you describe the intended context of use for your organochip technology in studying CIPN? Is it primarily for drug screening, mechanistic study, or both? Well, both actually, because the main application for the main application is for internal company decision, meaning that if you have a go no go to see if the chemotherapy you are currently developing will induce pain into the clinical trials. So it's more gathering data on various compounds that you are using, so either discovery or in preclinical, to see if you can move towards clinical trials, so it's a go-no-go decision, or to change the composition a little bit of your compound, or adding something that will lower the pain level. So basically, it's to help determine the most tolerated treatment for each use case. I have a question. Is a tech transfer to our labs possible? Yes, definitely. So as you can see on the screen, we act in different ways. So you can sell the devices and train you to use the device internally, so you'll be totally autonomous. We also have a screening service in-house where we internalize and we do the screening for you. And we also can do co-development where you come with a specific need and you want to adapt to different cell types into your our devices, or a brand new application. We are very open to that. But yes, we work also as a device provider and also an analytical service provider. Okay, last question. Apart if you have some. For CIPN, is the CIPN model available with other cell types, motor neuron, donor cells, and or cell suppliers? yes, indeed, I haven't shown everything, but we have established motor neuron culture, and we are working with most of the major cell suppliers to provide joint protocols. So if you are interested in a specific cell types or co-culture cell types, not only neurons, please feel free to ask us. I guess we have 24 protocols right now, so feel free to ask us. Okay, I guess questions are over. Thank you very much for your time. I hope this was helpful. Feel free again to contact us on contact@netry.com or visit directly our shop online to buy the devices, and we'll be more than happy to welcome you for a question and to welcoming you to the Neurofluidics community. Thank you very much, and have a very nice journey, afternoon, morning. Bye-bye.